Hello, everybody. Thank you so much for joining us today for this educational symposium. I'm delighted to be part of this uh, incredibly useful and educational experience. It is for me. Every time I listen to some of these talks from my colleagues, I learn a lot. So I'm going to talk to you today about gut inflammation, inflammation in Parkinson's as we understand it, the role that inflammation in your body might be playing in terms of underlying mechanisms of progression, perhaps pathogenesis, and I'm happy to answer questions at the end of the presentation. Here are a few disclosures with regards to some collaborations, consultancy. The questions that we're going to address today are here. What causes brain inflammation? Is there evidence of inflammation in Parkinson's? Are there certain types of cells or proteins, genes, hormones that might contribute to this inflammation? How does good gut health program benefit us? And what is the best way to keep inflammation in check? So I'm going to use this diagram that seems complicated, but really the message here is that the brain communicates with all external organs. We know that the brain sends messages to other organs, and that there are cells and there are tissues, and then there's the whole organism. And this communication between the brain and your external organs is very critical even for brain health, because these tissues are making molecules and metabolites that are communicating back to the brain. And so this bidirectional communication between the brain and these tissues is really important for overall health and brain health. And that was probably something that we didn't appreciate for a while, because we thought the brain was immune privileged and there was nothing that could really damage it other than things in the brain. But it turns out that that's not exactly right. Because it's in communication with other organs, what's happening in other organs may very well affect the brain and vice versa. Now, because of infectious disease, things like COVID-19, for instance, we have, as a species, evolved with strong evolutionary pressure for genes and gene variants that give you good and robust immune responses, so that you don't pass on infections to other people in your groups or in your tribes. And so the idea was that you could get to reproductive age, pass on your genes, and that was it. However, because of increased longevity, we are now understanding that this repeated exposure to infections and things in your environment, we call that chronic antigenic load. This combines with something we called inflammaging, which is low-grade inflammation everywhere in your body that happens as you are aging. And this aging of the immune system, which we call immunosenescence, these two things come together and contribute to age-related disorders like Parkinson's and Alzheimer's. We know that age is the #1 risk factor for these diseases. Now, with this schematic from my colleague, Dr. Okun, we know that Parkinson's has come a long way in its diagnosis and treatment, and that we're very hopeful that every day we understand more and more what might be the trigger, what might be promoting progression. And we know that Parkinson's, people with Parkinson's live very active lives, and they also have off time and on time, and that at some point they may experience, the typical motor symptoms that lead to a clinical diagnosis. And we want to understand how you transition from those stages and what might be interventions at each of these points. And you will hear from others today, about medications and other things like that. My lab is focused on understanding the non-motor symptoms of Parkinson's. And this schematic here kind of makes the point that we know that as age goes on, right here from -20 to your diagnosis, to 20 years beyond that, we know that the number of dopamine neurons in your brain is declining as you age. That's for everybody here. And for some people, that threshold may come sooner and or may come later. And we want to understand what are some of those things that accelerate that decline that may give you increased risk for Parkinson's development. And we know that about 2 decades before your clinical motor, your clinical diagnosis that is primarily diagnosed with motor symptoms, that 2 decades before that, we have things that are happening that are non-motor related and that may precede a lot of the motor symptoms. And we want to understand what are the earliest clues about that, and how does the immune system, which is the sentinels in the body, they're like the parents taking care of the dependent neurons that can't do much for themselves. How does the immune system in the brain and elsewhere give us clues about people who might be at risk, or how it progresses and you eventually lead this cross this threshold? So we believe that this stage here, 10 years before your clinical diagnosis, is where we have the perfect storm for Parkinson's development, maybe as early as 20 years. And it's a combination of aging, but aging of the immune system, which hasn't really been studied too well with its contributions to Parkinson's. It's an interaction with genes that you're born with, right? And the environment that you experience, your lifestyle choices and other things in the environment. And so this perfect storm may be happening here. And this is where, if we focus on these changes and how the immune system may give us clues about that, and inflammation may give us clues about that, we may be able to identify people at risk, and we may be able to identify people who already have the disease, who may be inflamed, and inflammation may be fueling their progression. So that's the part that we focus on. And this schematic makes the same point, that environmental factors can disrupt your immune function in your body, and it may alter expression of certain genes that we know are associated with Parkinson's risk. It may increase inflammation in ways that don't help your body or your brain. It may cause lead to production of autoantibodies and autoreactive T cells. And it can also directly affect tissues, right? And including your gut microbiome or neurons directly. And so all these three things in the middle contribute to systemic inflammation, which then leads to neuroinflammation, which is brain inflammation. And many studies have shown that vulnerable populations of neurons will then perish and start malfunctioning and then perish. So you all know about this idea that these diseases like Parkinson's may be proteinopathies or diseases of protein aggregation, right? That when protein gets gummed up, like the plaques and the tangles or the synuclein aggregates, that it may trigger activation of immune cells, right? And that's easy to understand if someone has a mutation, those rare mutations that lead to familial Parkinson's or familial Alzheimer's. And that cascade of inflammation will always be there because you can't get rid of that mutation, right? Just yet. And so that cascade of inflammation promotes neuronal cell death for vulnerable populations, and then you get different diseases depending on what parts of the brain are affected, right? But what about the 90% of individuals that have sporadic forms of these diseases, right? How can we account for that continuous protein aggregation or protein aggregation trigger, if you will, that then leads to microglia, which are the immune brain cells, activation, and then the neuronal cell death? Well, we and others have proposed that one way to get to this same cascade is through environmental exposures, chronic systemic inflammatory disease, perhaps peripheral infections, unhealthy diet, physical inactivity. So things that you do that affect inflammation in your body, or conditions that you have that affect inflammation in your body outside your brain. So this would then create the same trigger, the same cascade. And so this is a model that we have proposed and are testing, as are many other labs, to see if there's any truth to this, right? Now, there is a lot of evidence from different studies, brain autopsy studies, genetics, PET scans, biofluids, and epidemiology, supporting the idea that inflammation in Parkinson's plays a role at all different stages. We need to understand, when is it helpful, when might it be detrimental, right? And today I'm just going to focus on gut microbiome and the idea that gut inflammation may be one of those processes that may be fueling the progression or perhaps even the beginning of the disease. So you all have heard about synuclein, and synuclein is sort of, the big bad protein that misfolds in Parkinson's, analogous to, say, beta amyloid and amyloid plaques in Alzheimer's. And this protein is very critical for protecting those neurotransmitter vesicles that your neurons make to pass on signals to another neuron. So we have to have it, right? But what it turns out to happen is that when we get too much of it, it starts to misfold and it starts to aggregate, right? And so this aggregation is something that is believed to be one of the triggers for this proteinopathy, right? So a lot of effort has been placed on finding the nuclein levels. Are they low? Are they high? Trying to prevent its aggregation, trying to dismantle the aggregates, and trying to figure out, does that get you anything, right? And one of the things that has been proposed is that perhaps some of the aggregation doesn't start in the brain, that perhaps it starts outside the brain. For instance, Brack and his colleagues proposed that it may start in the gut, right? And this pink schematic just shows that if they take brains that have synuclein aggregation post-mortem, they can sort of plot a trajectory where they hypothesize that synuclein starts aggregating peripherally, it then goes into the brain, and it slowly works its way up into the neocortex, right? So this idea is that perhaps it starts in the gut, and it may very well be something that takes decades, but eventually it gets to the brain. Now, there's competing hypotheses that say some people have brain first and some have body first. It's really not that critical as long as we understand what your disease is about. And so there's a lot of effort for personalized medicine, for trying to understand exactly what kind of features you have and what kind of interventions you might respond to, right? But suffice it to say that the non-motor symptoms that we know that may start 2 decades earlier include GI dysfunction, right? And that supports the idea that maybe it is true that synuclein aggregation may start in the gut for some people. So the non-motor symptoms include GI dysfunction, and we know that most of the time they precede the motor symptoms. So we and others have proposed this idea that perhaps intestinal inflammation may be what's driving this synuclein aggregation in cells in the gut. The cells in the gut have synuclein, and then it propagates up into the vagus, and the vagus nerve, which is the highway that connects the brain to the gut and many peripheral organs, may be that conduit for this aggregated protein. But it could also just as well go through the bloodstream and the circulation and get into the brain in many different ways, or perhaps it doesn't happen at all, right? So it's a hypothesis, it's being tested, and we need to model it in different preclinical models. Then we need to see, is there any evidence in human populations that there's any correlation or any relationship to any gut health? And so about 10 years ago, we proposed this idea that inflammation was probably sort of this dark passenger of Parkinson's disease, that it was playing a role all the way in that Brack highway, from the gastrointestinal dysfunction to depression, sleep disturbances, motor deficits, and cognitive deficits and psychosis. And so again, it is a target that we have chosen to follow, chronic inflammation. And within that, one particular cytokine is tumor necrosis factor, or TNF, and I'll tell you more about it shortly. So if brain health is connected to gut health, then how does that happen, right? So this is a schematic of your gut. You have the gut lining, and in the gut lining, you have trillions of bacteria living there. And those bacteria you need to absorb the food that you're eating, to distinguish between toxin and non-toxin, and to keep a balance of healthy barrier function. When that is working well, these cells are very tightly closed together and nothing leaks in there that shouldn't. And so these immune cells down in the lamina propria or underneath are just doing their job and keeping the house clean. But when you have bacteria that are not so adaptive and they're more like pathogenic, that balance breaks down, and then you get leakiness between these intestinal epithelial cells, and you get this underlying inflammation from the cells that are below, and you get dysbiosis and inflammation, right? So let's talk about the microbiome and that bacteria that I just talked about. There's a lot of different microbiomes, and that just refers to the bacteria at barrier sites. Any site where you have contact with the outside environment will have a microbiome. So the skin has a microbiome, the mouth, the lungs, the urogenital tract, the stomach, and the intestines, right? And for the sake of this talk, we're going to talk about the gut microbiome. And where does the gut microbiome come from? Well, it comes from birth. The fetus is traditionally sterile, and the bacteria are acquired at birth as you go through the vaginal canal, okay? And this then gets shaped as the baby starts eating its own diet, mother's milk, its own diet, and then its exposure to the environment. And most gut bacteria in the microbiota benefits you, as I said before, because it is critical for food digestion, for wound healing, and it regulates immune cells in the gut that are in communication with brain immune cells, okay? Now, what happens when you take antibiotics is that you wipe those out, right? And sometimes people say, oh my gosh, I had terrible diarrhea from this antibiotic. Well, that's because you are not able to absorb the water from the food, and basically you have a imbalance in the bacteria that help do that. When this happens and you do it too many times, you leave a space for more pathogenic bacteria to take over, okay? And one of the things that happens when you get something called Clostridium difficile is that you could die. It's a life-threatening infection that needs to be taken care of or you will die. And so fetal microbiota transplants have been done to recolonize the immune system in the gut of people with these infections, and it is life-saving. So that's how we know the gut microbiota is important. The gut microbiome has been demonstrated to play a role in a lot of these conditions, including PD, but we don't know exactly which bacteria may be important to increase or decrease, right? And how do we know about it? Well, the bacteria taxa can be identified by sequencing the DNA. So that's what people do. They sequence the DNA and they figure out what they are, what bacteria are in there, and what they're making. And so remember that schematic I showed you at the beginning where I said communication between the brain and external organs. So it's now become a little bit more complex. It's the brain, the blood, the gut microbiota, where all of these sites are making important chemicals to communicate with each other, okay? And so how do we know that this goes wrong in PD? Well, there's now been over 18 studies showing that people with Parkinson's have gut dysbiosis, or an imbalance in the gut microbiota. There seems to be some agreement that these bacteria are increased and they tend to be more opportunistic pathogens. And you might say, well, is this something that could be fixed with probiotics? Well, that is under investigation in different labs. It could also be modified by diet, potentially. My colleague, Carly Rush at the Fix Cell, has been doing some beautiful studies looking at the effects of Mediterranean diet on gut microbiome in PD. There's also some understanding that there are some bacteria that are decreased, right? And these are shown here. And importantly, these bacteria are those that produce something called short-chain fatty acids. And short-chain fatty acids tend to have an anti-inflammatory effect. So if you lose the bacteria that make them, you may make the microenvironment in the gut more inflamed. So a graduate student of mine in immunology at Emory Madeline Hauser, who's now in a faculty position there, ran a study in collaboration with Heidi Payami at UAB, where we took the stool of individuals that had their gut microbiome sequenced, and we extracted the inflammatory cytokines from that stool. Okay, so we had spouse controls and we had people with Parkinson's, and we found 4 factors: interleukin 1 alpha, 1 beta, something called C reactive protein, and CXCL8, which is the chemokine that attracts immune cells. We found these 4 to be different in people with Parkinson's versus their spouses. Okay. We also found in another study in Finland, in collaboration with Phil Shepherd-Jans, that the same happens, same findings in a different cohort. So it seems that these relationships are being replicated in different groups of people with Parkinson's. In this study, we also measured these short chain fatty acids, and we looked at plasma as well as stool. And very interestingly, we did find that people with Parkinson's have lower short chain fatty acids levels in their stool, as shown by butyrate and propionate, and they have higher levels of something called calprotectin. And calprotectin is sort of the quintessential biomarker for inflammatory bowel disease, suggesting that there's a leakiness in the gut in at least some of the patients that we examined. Another thing that we found is that a correlation, okay, with higher levels of the short chain fatty acids and lower levels of inflammation seem to correlate in this cohort with later onset of Parkinson's symptoms, implying that if you have higher short chain fatty acids, lower inflammation in the gut, you may end up with later manifestation of symptoms. Okay, so these findings probably need to be replicated in other cohorts, and to show directly the power of short chain fatty acids, we definitely need to run clinical trials, because this is merely an association. So what is the best diet intervention for good gut health if you have Parkinson's? Everybody always asks me that, and I'm not a nutritionist. But my colleagues, Lori Mishley and Carly Rush, have really good resources that I would encourage you to look up for how nutrients and risk factors for PD may affect progression, and sort of practical things you can do in your diet to have more healthy eating behaviors. And down below is Carly's paper on the Mediterranean diet in people with Parkinson's, in its ability to reduce constipation, and changes the microbiota after a 5-week single-arm pilot study. So going back to the hypothesis that gut inflammation may contribute to misfolding of proteins that tend to aggregate and may contribute to this proteinopathy, we want to examine the idea of inflammatory bowel disease. Is there a relationship? And so inflammatory bowel disease, Crohn's, for instance, has genetic predisposition. There's environmental factors that increase your risk for it. And there's a disease initiation where it's not clear what the trigger is. It could be infections, it could be drugs, but there's lots of that tight junction and epithelial integrity in the barrier that I told you about, and you basically end up with the commensals, or the good bacteria, losing out to other more opportunistic bacteria. And then you have this expansion of autoinflammatory processes, and antimicrobial antibodies start to circulate, and that's the subclinical stage. Then you end up getting your IBD diagnosis when you actually have bowel damage, tissue remodeling down below the epithelial layer, and this very uncontrolled immune response, right? And people with inflammatory bowel disease and other autoimmune types of things, not all IBD is autoimmune, but when you have IBD, people tend to get on anti-TNF drugs, or Enbrel, Remicade. You've seen them, the commercials. They are aimed at targeting the inflammation in the gut, right? So why do we think there might be a connection, right? In addition to what I just told you about, perhaps the dysbiosis in the gut and the inflammation driving the synuclein aggregation. Well, there's interesting studies, although they're small in the number of patients that they examined, that patients with IBD are more likely to develop Parkinson's. However, if you take the people with IBD that have been treated with anti-TNF therapy, their risk for PD is now reduced by 78%. So again, it's epidemiology. It's not to imply causation, but to help you appreciate that there might be an association between strong inflammatory conditions in the gut and later risk for Parkinson's, unless the inflammation is treated. So this TNF, or tumor necrosis factor, is something that immune cells make in the brain, in the circulation, in your gut. And it's important for many different things. People know that it promotes inflammation. It's the cytokine that's elevated in rheumatoid arthritis and psoriasis and ankylosing spondylitis and a lot of different autoimmune conditions. What a lot of people don't know is that it's also important to fight infection, right? And it goes up as you're aging with chronic diseases, diabetes, metabolic syndrome, obesity. You have a lot of TNF circulating in your body. What people don't know, though, is that it's made first as a protein that's attached to the cells, attached to the cell membrane. And in this form, this TNF is really important for you to fight infection. It's also really important for myelination, or the sheath around nerve axons that transmit impulses between cells. However, this protein gets clipped by a protease in the cell so that it now is soluble or circulates in your blood and goes elsewhere to have its effects. And in this form, it's pro-inflammatory and it promotes demyelination, okay? So one name, two different ligands, two different proteins that have very different biologies, okay? So why is this important? Well, because when people started targeting and making drugs for tumor necrosis factor, the easiest way to cover it or to neutralize it or to block it is to try to block the production of it or to block its receptors, right? And you have to appreciate that the transmembrane form is good because you need it to fight infection, and the soluble form is potentially bad if you have it in excess because it promotes inflammation and tissue destruction. So what the antibodies and decoy receptors do that are the FDA-approved TNF inhibitors on the market is that they block both the membrane-bound and the soluble, right? And they don't distinguish between these two. So people that have Crohn's and RA and psoriasis and take these drugs can control their inflammation, but they're open to opportunistic infections like tuberculosis. And during COVID, they really had to hide because they were at higher risk. So the idea really is it's better to just find a way to block the soluble or the bad ligand instead of both, right? And so all TNF inhibitors block both and they don't distinguish. So there's a liability for, more demyelination. So you get these MS-like symptoms, and it's immunosuppressive because you can't fight infections. So the better approach is to just block the soluble, okay? So before I started my academic career and I was in biotech, I was fortunate enough to be part of a team that developed this new modality, new biologic, new TNF inhibitor, which because of its mode of action only neutralizes the soluble form of TNF. Why? Because it sees it and it thinks that it's self, and so it forms these mixed trimers here, and it sops up all the soluble TNF away from the blue receptors, okay? But it can't do this. It can't exchange when the TNF is stuck to the membrane. So it's 100% soluble specific, okay? So the idea was give them to people with rheumatoid arthritis, give them to people with IBD, and it's going to be, perfect. So we did a lot of studies. There's now been over 65 papers published in different labs, not just mine, in different disease indications, showing that soluble TNF inhibition can decrease inflammation, increase neuroplasticity, can be neuroprotective without being immunosuppressive. And the Alzheimer's Association funded a trial in Alzheimer's disease for people with inflammation. So that's how they were recruited. You had to have high C-reactive protein, high sedimentation rate, high hemoglobin A1C, or ApoE4, right? And they were given this drug subcutaneously once a week for 3 months. And then they looked at markers of inflammation, right? They're not monitoring cognition because it's a phase 1b safety trial, just to see if there's any, ability to dampen the inflammation. And they looked at MRI because MRI gives you an indirect way to look at inflammation in the brain. They looked at cerebral spinal fluid markers of inflammation, and they looked at gray matter cortical disarray measurements. Okay, so just to show you that the high dose of this drug, it was an open label, reduced a lot of markers of inflammation in the CSF. So all these are inflammatory cytokines and chemokines that were reduced after 3 months of the drug. And the correlation with the MRI free water, which is an indirect measure of, say, water for swollenness against around axons, was highly correlated. So the high dose of the drug seemed to have reduced inflammation by 2 different modalities. Then they looked at white matter integrity. And the white matter integrity is something that they use something called apparent fiber density to measure, okay? And it's basically looking at the white matter tracts that are, myelinated, that carry the impulses in a place called arcuate fasciculus, which is critical for Alzheimer's in memory consolidation. And they found that after 3 months, the soluble TNF inhibition was able to strengthen these axons and make them more cylindrical, more tonic, and less swollen. And as a comparison, so that's the green group, 10% increase in white matter fiber density. As a comparison, the ADNI database of 36 participants which did not receive drug, their white matter integrity declines over this time, right? So it looks promising. It looks like it may be good to perhaps, boost the axons that are there and to reduce swelling. So I'm going to summarize and tell you that for this part of the talk, then, there's overwhelming evidence that inflammation is involved in aspects of AD pathology as well as PD pathology. It's our work, but many other people. And this phase 1b proof of biology study in Alzheimer's patients showed that the biological changes that we've been measuring in these preclinical models with the soluble TNF inhibitor are the same in animals and humans. So it's encouraging, and the next step clearly is to determine whether these changes in biology can lead to clinical improvement in terms of memory, right? A phase 2 clinical study is launching now, it's enrolling now, and a trial in early PD is under consideration. And so we're very encouraged that by understanding how immune cell aging and chronic inflammation resulting from this gene environment interplay and representing the perfect storm could be a target for risk identification for development of anti-inflammatory immunomodulatory therapies that can arrest, if not prevent, the progression of disease. We know that gut health is important for brain health. We know that probiotics may change the gut environment and reduce inflammation, and they'll make you feel good. So far, there is no solid evidence that it can slow down your Parkinson's, right? But if you feel better by taking these drugs, if you have a good diet and you are able to lead a more productive and happy life, then by all means, right? We are definitely thinking along the lines of using gut health measures as endpoints for clinical trials and in interventions that could inform on that, because it seems to be a very important part of people's lives. And in humans, we know that lifestyle choices, diet, exercise are all likely to affect this G times C interplay, gene by environment interplay, and to impact gut and blood-brain barrier permeability. So if you make bad choices in terms of lifestyle, if you're physically inactive, you will be more subject to leakiness of things into your blood and across your gut that shouldn't be there, right? And that slowly but surely influences your risk for these age-related diseases. And so finally, the mitigation of chronic inflammation and chronic systemic inflammatory diseases, which are so endemic in our communities, may reduce the risk for PD and perhaps AD and their progression. And so to give credit where credit is due, the work that I presented to you on the inflammation in the gut and microbiome was done by Madeline Hauser, who was an immunology graduate student and now is a research assistant professor there in the Department of Nursing. And every person listed here, most of the work was done at Emory. We continue to investigate the role of the gut-brain axis in collaboration with Michael Okun and Nick McFarland through the Parkinson's Foundation Research Center of Excellence. So we're collecting blood and we're collecting colonic biopsies and we're collecting stool for people with Parkinson's, controls, and people with IBD so that we can understand what are the commonalities between these two diseases, and is there really a solid base, mechanistic base for associating the two things with risk. So if you're interested in that, please contact us. We're still enrolling at the Fixed Cell, and we're also enrolling people with known genetic mutations that increase your risk for Parkinson's, like LRRK2 and GBA. So we're always there. We cannot do this work without you, and we thank you for those of you that have enrolled and participated in our clinical trials. I want to give thanks to the Fixed Cell Institute as well as the CTR&D for the support for all this work, as well as the Michael J. Fox Foundation. And I'm happy to answer questions. Okay, is there any linkage between constipation and urinary incontinence? So in our Hauser movement disorders paper, we had a questionnaire where we asked about GI dysfunction, sleep problems. I don't recall that we asked about urinary incontinence, but we may have, and I can look back, or you can look it up. But you're welcome to contact me, and I will look at that. There is definitely a connection to the kidney and important proteins that are important in the kidney and in the urinary tract with regards to something called LRRK2. And I think that, inflammation affects all tissues, and it will create dysfunction in ways that are very subtle. We also know that urinary tract infections make everything worse. And we suspect that that is a function of the inflammation that circulates and that affects then your brain and all the other circuits. Are there recoverable symptoms? Hyposmia, regaining smell. Okay, so recoverable symptoms. So my understanding, and Michael, if he's there, can pipe in, or any other neurologist. My understanding is that the hyposmia and anosmia and loss of smell that patients experience is not Parkinson's specific. It's also happening in individuals that are at risk for Alzheimer's disease. And I don't believe that it's a reversible symptom, but by itself, it doesn't mean that you have AD or PD, right? It is part of a constellation of symptoms that make part of a profile that helps a clinician make a diagnosis, along with the very important neurological exam for balance, posture, rigidity, stability, that kind of thing. So as far as we know, the hyposmia that you're experiencing there is not a reversible situation, unlike some of the loss of sense of smell or taste that people experience with COVID-19, for instance.
So Dr. Tansey, that was fantastic. Amanda tells me that we have to stick to our schedule, that we can take questions. And if there are more on the chat, we can bring them to
answer them.
Ask the Expert panel. And if we can't answer them and we need Dr. Tansey's input, we can get back to people.
Fantastic.
So one of the questions here is, a lot of over-the-counter probiotics are heavy on bifidobacterium and lactobacillus. What does that mean for Parkinson's disease patients?
Yeah, that's a great question. So bifidobacterium and lactobacillus is one of those bacteria that seem to be increased in PD. And, people say, well, then why should I take more, right? It's a bad thing. But I would say that we do not know that, because some of the changes that you're seeing in the microbiome maybe what we call adaptive changes. They're changing to protect you. So it's perhaps that it's a response to the, situation that these families are increasing. And what we're doing now with funding from Fox Foundation and the Brin Family Foundation is we're actually testing individual bacteria in organoids to see exactly which one's good, which one's bad, right? We can't just say, oh, because it's up in PD, it must be bad. It may be going up to protect you or for a reason, right? And so we can't tell you right now, don't take it because it's a bad one or because it's already up. Like I said before, if talk to your doctor, and if it changes and improves your brain health or your gut health, then I wouldn't change.
And I think the second question is in a similar vein. There are many advertisements for Solase, parentheses, PS128. Is there any evidence for its effectiveness?
Yeah, Coles. So things that again improve your gut health, I think, are underrated. And if they help you improve and be more regular, and feel better, so that you can walk more or exercise more, I think that's really the key. Um, I wouldn't want you to spend a lot of money on the expensive probiotics that need to be refrigerated and this and that. Personally, I think the, over the counter CVS pharmacy 50 and over are awesome, but it's different for every person. So I would say talk to your doctor and find what's right for you. Absolutely.
Thank you so much,
Yeah.
Dr.
Thank you
Tansey.
so
We're
much.
so grateful that you made
Okay.
the effort to be with us today.
No problem. Thank you so much.